Professor Matthew Gentry obtained his PhD from Syracuse University under the mentorship of Dr. Richard Hallberg. He is currently the Antonio S. Turco Professor of Molecular and Cellular Biochem at University of Kentucky. Of relevance to today is his directorship of the Brain and Epilepsy Alliance for Metabolism Research and the directorship of the multinational NINDS-funded Lafora Epilepsy Cure Initiative. Today, he will speak on Lafora disease, and the title of his presentation is Preclinical Treatments for Lafora, a Childhood Dementia Driven by Perturbed Metabolism. Thank you very much.
Thank you very much for the talk. Can you hear me okay?
Yes, we can hear you well. Thank you.
Great. And feel free to, what I'm going to show today, take pictures or record. No problem on that front for this work. So thank you very much for the introduction. And I want to thank Chris and the organizing committee for the opportunity to speak and just really acknowledge what the childhood dementia group there in Australia has done. It's a really impressive effort. And I just want to applaud the vision and foresight that you all have had to bring these groups together. So, there we go. So first, I need to give you a quick glycogen metabolism refresher. I won't drill down into the details of this, but glycogen is the way that our cells store sugar, glucose. It's done by utilizing two enzymes, glycogen synthase and glycogen branching enzyme. Once it's stored and needs to be recycled back into glucose, there's really three enzymes that are important for that: glycogen debranching enzyme, glycogen phosphorylase, and then this enzyme laforin that we'll hear a little bit about today. Once glycogen has been made by the cell, glucose comes in via different transporters depending on the tissue type. It's converted into glucose 6-phosphate and made into glycogen. It can be then released from the glycogen and go on to drive all of these cellular processes and impact all of these different types of metabolism in the cell, and really impact things on the physiological level, such as aging, dementia, and cancer. Now, if there are perturbations in the enzymes that are involved in glycogen metabolism, and there are more than just these are the sort of the central five, but there's about 15 or 20 different key enzymes involved in glycogen metabolism. And if you have a mutation in those, you end up with a glycogen storage disease, or a GSD, and these impact about 1 in 20,000 individuals. The most popular or probably famous of these is Pompe disease, which was the story of it is told in this movie, Extraordinary Measures here. So the glycogen storage disease that I'm going to talk about today is Lafora's disease, and it's also a childhood dementia. It's a fatal autosomal recessive disorder, and these individuals develop normally for about the first 10 to 15 years of life, and then they oftentimes will present with a myoclonic event. This is an event like you see here in this beagle dog that has Lafora's disease. This is a pretty strong case of myoclonus. A myoclonic event is like when you fall asleep at night and your body jerks. That's a myoclonic event. But these patients will have those events over and over repeatedly, like you see in the dog here. After those myoclonic events, the patients have epilepsy. Those epileptic episodes increase in both number and severity over the next 5 to 10 years. Late teens to early 20s, there is a stage of childhood dementia that is quite rapid with horrendous epileptic episodes, and then the patient usually dies in their mid to late 20s. Now, the hallmark, the cellular hallmark of this disease are these pink aggregates that you see here that are stained with puritic acid Schiff or PAS stain, and these are referred to as polyglucosan bodies. They can also be referred to as corpora amylaceae or Lafora bodies. I'll probably refer to them as Lafora bodies throughout the talk here. These aggregates of aberrant glycogen are found in the cytoplasm of cells from almost every tissue in these patients. So they're in the cardiac tissue, the muscle, and in the brain. And it's really the brain where we see the massive problems of neurodegeneration that drives the epilepsy, the dementia, and really the death of the patient. So a summary of this disease for the last 110 years or so, it was first described by Gonzalo Lafora in 1911. And then in the late 90s and early 2000s, groups at UCLA and University of Toronto identified the 2 genes that are mutated. So to be suffering from Lafora's disease, you either have mutations in both copies of your EPM2A gene that encodes laforin or your EPM2B gene that encodes malin. After the identification of those genes, really for the next about 20 or 25 years, what we've been focused on is defining tools to study the disease. So biochemical tools, cell culture models, and mouse models, as well as structural biology, to really understand the underpinnings of the disease and how perturbations or mutations in these 2 genes drive disease progression. And so that has led to a period of excitement now that we've built up enough knowledge where we have, in fact, developed 3 therapeutic platforms and I'll tell you about one of those in detail today. And this work, starting in 2016, was funded by a large grant from the National Institutes of Health that brought together an international group to work on this problem. And we've also had funding from Cure Epilepsy and the Epilepsy Foundation. So the NIH grant brought together all the groups that you see logos listed here, the different individuals from those different institutions. And really what we've been able to do is define the basic mechanisms of the disease, figure out what's causing it at the cellular protein level, and then attack that problem with different therapeutics. And in each case, what we're really trying to do is remove or stop the progression of these Lafora bodies. And I'll walk you through a couple of examples of that. So mutations, as I said, in either the gene that encodes laforin. Laforin is a glycogen phosphatase. It removes phosphate from glycogen. Mutations in that gene lead to aberrant glycogen metabolism, Lafora bodies, and that drives the disease. Mutations in malin, which is an E3 ubiquitin ligase, also goes through the same path. And this is what we've shown over the last sort of 5 to 8 years with using those cell and mouse models of the disease. So we do in fact have really outstanding mouse models of the disease. This is a wild type brain, a Lafora knockout mouse brain, and a malin knockout mouse brain stained with that PAS staining. And you can see the pink aggregates of the Lafora bodies in these mouse models. The mice do have a higher propensity for seizures, and they also undergo neurodegeneration, but they do not die at a younger age. So it's not a perfect model, but it's quite good. We also have an antibody that detects these Lafora bodies, and I'd like to show this because this shows just how many regions of the brain are impacted by the disease. It's really every region of the brain, the hippocampus, the cerebellum, the cortex, really every region of the brain is impacted by these aggregates. And so our unified goal of the Lafora Epilepsy Cure Initiative is to either stop the progression of these Lafora bodies or to degrade them once they have already formed. And the reason that we're focused on this is because using different genetic mouse models, what we've been able to show is that a 50% reduction in glycogen synthesis eliminates the Lafora bodies, and this then greatly reduces the epilepsy, and it eliminates the neurodegeneration in the mice. So this is a sort of a rare example in the brain anyway, where the aggregate in fact is driving disease progression. And so that's why we're focused on these aggregates. And so to think about this in just sort of cartoon form, the way we think about glycogen in the brain is that you have normal synthesis of glycogen and it builds up, and you have normal degradation, and that is used to drive metabolic processes in the brain. And these are the enzymes that are making and breaking down glycogen in the brain. In Lafora's disease patients, they can make the glycogen-like aggregates, but they can't break it down. And so you get this perturbed metabolism. So our goal then is to reduce that level of those aggregates, release that glucose back into the system, and hopefully the hypothesis is that we would rescue this metabolic phenotype. And I'll in fact show you work today that we've been able to do that in mouse models. Okay, so this sort of summarizes where we stand in our consortium. We've generated really 3 platforms that target these Lafora bodies. That's an antisense oligonucleotide. This is an RNA-based therapy targeting glycogen synthesis. It's similar to what the previous speaker spoke about regarding mRNA and siRNA. So this is an RNA-based therapy. I'll tell you today about an antibody-enzyme fusion. and its work. And then we also have a small molecule that inhibits glycogen synthesis, and it stops Lafora body formation as well. And then we're also looking at repurposing drugs that target inflammation or other pathways to sort of fill the gap while we develop a more disease-modifying therapy. And then, ultimately, as some of the speakers have said, we'd love to see gene therapy be utilized, but the reality is that that's not in the immediate future as far as we see it. So what's an antibody-enzyme fusion? If you're familiar with the cancer community, they utilize antibody-drug conjugates quite often. These are antibodies that target a specific type of cancer using an antibody fragment, and then they attach a cytotoxic drug to that antibody. The antibody then takes the drug to the cancer, and the goal is to kill the cancer. Well, an antibody-enzyme fusion is a different spin on this, and a company called Parasail has identified a fragment of an antibody or a Fab that can gain access into cells via this ENT2 transporter. And the beauty of this is that it gets directly into the cytoplasm without going inside of a vesicle and without the pH being largely changed. It can be a neutral pH. So what Parasail has done is shown, and in collaboration with other groups, that this antibody fragment can be fused to different enzymes, and those enzyme fusions then can get taken into the cell. So what we did was identified a enzyme that can degrade Lafora bodies, this AMI2A. This is pancreatic alpha amylase. It's a human enzyme. The antibody here, the Fab, is humanized, and so this is a fully humanized fusion, and the name of it is VAL-0417. So the goal here, or the hypothesis, is we could infuse this VAL-0417. It would get into the cells. Once inside the cytoplasm, it would degrade those Lafora bodies, and then the glucose could be utilized in metabolic processes. So I'm going to skip a lot of the work that we've done because most of it has been published, but we were able to show that this VAL-0417 is successful at doing this in skeletal muscle and in cardiac muscle, but this is a brain disorder. And the problem is that this is a large fusion protein and it can't cross the blood-brain barrier. So we had to come up with a way to administer it directly into the central nervous system. So we tested both intrathecal as well as intracerebroventricular administration. These are both used in the clinic for different types of drug delivery to date. And so we tested this in mouse models. And what we observed was that we could get drug in to the brain, into different regions of the brain via each method. So what we looked at after administration, we sectioned the brain into 6 different sections and just asked how much drug was in each section. This is what we saw by intrathecal. Here's our PBS or saline control, and this is the ICV administration. So ICV clearly got more in, but intrathecal also got a good amount of drug in. I like to show this figure here because it really exemplifies how important that Fab is to make this happen. So if you just focus on this panel right here, the difference is this is introducing the amylase by itself via ICV administration into laforin knockout animals. You see very little brown staining, and that's indicative of the enzyme getting into the cells. Very little cellular entry of the amylase by itself. But when we attach that Fab, and now we have VAL-0417, you see a lot of brown staining. So it's able to get into the cells, into the cytoplasm where the Lafora bodies are. So here's really the key experiment. So what we did was we took Lafora knockout animals that were about 7 months old. These animals have very robust pathology, lots of Lafora bodies. We administered VAL-0417 to these animals for 7 days. They get a continuous administration by ICV. We euthanized the animals on day 8, and then we did a battery of tests, but I'm just going to show you that past staining because it's quite dramatic and it's indicative of all the other results that we got. So here's what we saw when we stained for those Lafora bodies. These are the Lafora bodies in the untreated animals. So these animals had no brain surgery at all. Here's the Lafora knockout animals that had brain surgery, but then were infused with PBS. You see the Lafora bodies throughout the image here. Then strikingly, when we treated animals with VAL-0417, the Lafora bodies were completely gone. We struggled to find any in these animals, and the tissue instead looked much like what we saw in a wild type untreated animal. And just so you can see, this wasn't in just one region of the brain. Here's examples seeing Lafora bodies in the cerebral cortex, the thalamus, the cerebellum, the brainstem. We also looked at the hippocampus. And in each case, there's really nothing to be seen down here regarding a Lafora body. So really, VAL-0417 ablates the brain Lafora body. So this is great, super exciting. But the Lafora bodies are gone. The next question is, can the glucose be utilized for cellular metabolism and rescue and change the metabolic profile of these animals? So to do this, to test this, we utilized metabolomics, where we can take a brain, pulverize it into a powder. From that powder, we can extract out the polar metabolites. These are metabolites that are involved in central carbon metabolism, things like TCA cycle metabolites, glycolysis, pentose phosphate pathway, etc. And then we can analyze them by mass spectrometry. We derivatize those. Once derivatized, each metabolite gives its own signature profile here of a peak. We can curate these data and then do these multivariate analyses where we're looking at sort of diseased versus control groups. And so that's exactly what you're looking at here. In gray, you've got the wild type animals. In red here are the Lafora disease untreated animals. Each animal is a dot. And this is a really data-rich plot because each dot is about 70 different metabolites. So what this image here figure tells us is that the metabolism of a Lafora disease animal is different than that of a wild type animal, which is what we expected. So now what happens if we infuse just PBS into these animals? So we're going to do brain surgery and infuse the PBS. And when we do that, it changes the metabolic profile, which is not unexpected. Because we're doing brain surgery on these animals. They have inflammation that changes the metabolic profile. But excitingly, when we infuse VAL-0417 into animals, what we see is that their metabolic profile now matches that of the wild type animals. So all 5 animals now look from a metabolic standpoint like the wild type animals. So the last piece of data that I'll show you here, it was a super exciting piece of data we got sort of the end of last year. So in addition to having a glycogen defect, what we've recently discovered is that the Lafora disease animals have what's called a glycosylation defect. So you have glucans are glucose based, glycans decorate proteins with different types of sugars. And these sugars are depicted by these sort of squares and circles and triangles. I won't go into the details of those, but what we can do is using a really high-powered imaging technique called multi-imaging is we can ask this particular sugar, where is it at in the brain and how much of it is at each location? So if it's red, that means there's a lot of it. If it's blue, that means there's not much in that space. So this is looking at a wild type mouse brain for this particular sugar right here. We can look at a number of different sugars. So here's 3 different sugars in the brain and where they are at. If we look at the Lafora disease animals, you're going to see a massive difference in the image here. We have this really very dramatic in-linked glycosylation defect in the Laforan animals. So now the question was, what happens if we treat the animals with VAL-0417? And to our really surprise was how well we rescued this phenotype. So this means that glucose that we're releasing, not only can it be utilized for central carbon metabolism, but it can also be utilized to restore appropriate glycosylation in the brain. So this is just to kind of summarize where we are. We've got this antibody-enzyme fusion. We've tested it in both malin and laforin knockout animals of Lafora's disease. We get equal results in both models. We're defining the minimum dose. To date, we know that we can go down to just 3 days of administration and that we still get that same maximal response that you see right here. So 3 days of administration degrades the Lafora bodies. And so we're currently determining sort of the minimum length of treatment time in these preclinical models, establishing human and mouse biomarkers, and we're ramping up to do a canine safety study in the coming weeks. And so I just want to acknowledge this was a really team effort. A number of people in my lab involved in this work, as well as Ramon's son's lab, who's a close collaborator of ours, our funding sources. And just in terms of thinking about, how this could be utilized and implemented for other childhood dementias. First off, I would say, what was key to us was getting that NIH grant to bring all of us former competitors together to work on defining the basic mechanism and then translating that into therapeutic platforms, that knowledge, and also working closely with the patient advocacy group. And then I guess the second piece would be that, any type of childhood dementia that has a glycogen or a glycosylation defect or perturbation could potentially benefit from the work that we're doing on Lafora's disease. I'm not familiar with all 70 of the childhood dementias, but I think that's something to keep in mind as this field develops. And so I'll stop there and just thank again the organizers for all the great work that they're doing to bring this group together.